Hi, everyone. Welcome to Foresight's Biotech and Health Extension seminar group. We're really excited to have repeating guests here today to hear an update on the Biomarkers of Aging Consortium. Many of you, I think, who are online right now, or of you guys afterwards watching it on YouTube, may remember that we've had a launch seminar a little bit earlier. And so if you want to go back to that, feel free to do so if you want to just have a bit more of an update on what this consortium is really up and doing. Today, we're going to hear an update on what you guys have been up to. We're really excited to have you all here. We're going to have a few folks presenting at the beginning, and then we have a Q&A for everyone who's in here right now and would like to ask some questions, propose some opportunities for collaboration. I'm going to be sharing more about you guys here in the chat. But yeah, without further ado, Vadim, please take it away, share your screen, and it's really good to see all of you here again. There's been some exciting changes, and we're very excited to hear about them.
Thank you, Alison. So we're going to have a brief update today on this consortium that you might remember we presented on it a few months ago. It was in the spring, I think, around March, probably, of this year. And a lot of things have changed since then. So I think it's time for an update. So what is the current status quo in the use of biomarkers? So it's been rapidly expanding field. And clocks are being published every day, nearly every day these days. And they're actively used in epidemiology, for example, for observational population studies, in basic research, in anti-aging, in rejuvenation. That's another rapidly developing area. And also in clinical trials. And so this is all very exciting, but there are many kind of questions remain. And so this slide shows the example of the clinical trials that already use or have used the biomarkers of aging, which is, of course, very exciting. But again, so many of the biomarkers are here, and exactly which to use and what is the best, or what exactly should be measured is not fully clear. So these are the questions that we typically get. I would say maybe daily even. What biomarker should I use? Okay, so what do actually various biomarkers measure? And how do we validate biomarkers? And what will be the progress towards regulatory approval of these biomarkers? There's a potential, the use of biomarkers is extremely important in my mind, because it could be actually a key factor in progressing towards longevity interventions, because in clinical trials, we have to use it. And if the biomarker is validated, perhaps it would really accelerate the use of and development of geroprotectors. With this kind of ideas in mind, we formed a consortium of biomarkers. This was in 2022 already. It's over 1 year old. I should say that I'm presenting here as a senior author, but it was actually conceived by a junior people, and primarily Madi Mokri, and also Jesse. They're here, and Madi will present as well. And these junior people self-organized and brought more senior people, and become really a vibrant community currently. So who we are? So we are currently a consortium of scholars and practitioners developing, validating, and implementing biomarkers of aging longevity. This includes academic members, faculty, mostly affiliated with leading research institutions and groups in the field of aging, as well as practitioners, biotech, members of biotech companies, and other people, non-profit organizations who are at the forefront of research and development in the longevity industry. So what we try to do, we try to facilitate development, application, communication, collaboration, sharing of data tools, and as well as information to establish reliable biomarkers of aging, particularly for the identification and evaluation of longevity interventions. We have a scientific committee, and here is the existing current scientific committee. Of course, it might change. It's all in progress. So myself, Mike Snyder, he was supposed to present today, unfortunately is unable today, and Vittorio, Andrea, and Eric. And this is scientific committee. This is the executive committee. These are the people who actually do the work. And this is Madi Mokri. He shares this position 50% in my lab and 50% with Mike Snyder at Stanford. Jesse, he's also a senior member of my lab. Alison and Dane. And of course, everybody is just such a pleasure to work with these people. It's really amazing. So we have regular meetings and it's really very nice. This is a current advisory board. This is Steve Horvath, Luigi Ferrucci, Brian Kennedy, Jamie Justice, and Steve Cummings. What this slide actually is not updated. So actually, Nir Barzilai also recently joined the board. We just have not updated the slide, but Nir is also there. We're of course very grateful for all the advices we get. In terms of the roadmap of this group, it was conceived the consortium last year at the Cold Spring Harbor meeting, and we first worked on the first kind of study developing consensus across different concepts, trying to define some basic terminology, brainstorm concepts, and analyze various use cases of biomarkers. This started in February 2023, and we've written a manuscript which has been published in Cell about 2 months ago. There is a second study which we worked over the summer on cross-population validation. This includes challenges and recommendations for robust cross-population validation. And this manuscript is currently under review in Nature Medicine. So that's another really, I think, important contribution to the field. We also have various other activities. For example, we plan a first symposium. It's a biomarker of aging symposium, which will happen at the Bach Institute on December the 4th. And we also work on the extension of the studies, study 3 on critical next steps for the field. You might say a few words on that as well. It says planned here. Actually, there is ongoing work on that. So for the first manuscript, we developed this asynchronized collaborative writing approach. So where there was a drafting team. The initial draft was prepared by Madi and Jesse. And then we invited the first group of scholars and extended the work, and then the second group of scholars, and then as well as senior members of the community, and back and forth with numerous rounds of revisions. It was really extensive work. And so for the senior kind of decisions, there was a group of Luigi and Steve and myself who made many decisions. And I would say everybody had to compromise. I myself was voted down a few times, it's okay. I think we work as a group, and I think it's a very nice community. So this is the first study that we published, biomarkers of aging for the identification of evaluation of longevity interventions. 25 labs contributed to this study. And again, this was very nice. So just very briefly, just tell you about this. First, we wanted to define some concepts, what we are actually talking about. For example, what is aging? What is biological age? And so on. So very roughly, we define aging as a process of accumulation of consequences of life, such as molecular and cellular damage, that eventually leads to functional decline, chronic diseases, and ultimately mortality. This is biological age that we described in this manuscript. Conceptually, it's individual's age defined by the level of age-related biological changes, such as molecular and cellular damage accumulation. And in practical terms, it's summarized as a number when units of time matching the chronological age where the average person in the reference population shares the individual's level of age-dependent biological changes. Of course, we cannot claim this is the ultimate truth. This is the current consensus definitions, and they might change as the field develops. And concepts like this, we have over 10 in the manuscript. There is a separate table on that. I encourage you, because there was a lot of brainstorming and revisions for all of them, all of these concepts. We defined types of biomarkers, for example, molecular biomarkers, which could be divided into omics and specific molecules. As for omics, there are biomarkers based on epigenomics, transcriptomics, proteomics, metabolomics, and there are also physiological biomarkers that define based on functional performance, physical characteristics, as well as digital biomarkers that include both wearables and non-wearable technologies. Another way to classify biomarkers is based on their use. For example, there could be predictive biomarkers, prognostic biomarkers, which deal with mortality, age-related functional decline, morbidity, and health span. There also response pharmacodynamic and based on surrogate endpoints, as well as discovery biomarkers. Again, there are many details on these types in the manuscript. I encourage you to read this work. So now I'd like to say that there are many challenges, and this is just arbitrary list of challenges that still need to be addressed. I will not go through them in detail. It's just there's a lot of work very at the beginning, I think. I'll just show one example from our own work in the lab on some of the challenges that we try to address. So, for example, this challenge is on the adaptive versus damaging conditions. And what this kind of refers to is that with age, we accumulate negative consequences of life, like molecular damage or byproducts or epimutations and so on. But then organism, as it accumulates damage, also finds a new homeostatic state and activates protective or adaptive responses. And these responses, they also have age-related changes. And therefore, when they are used to construct a clock, they would end up as part of the biomarkers. They are not necessarily aging per se, but kind of response to aging. And so it would be useful to really separate this component, because even though it may be used to quantify biological age in terms of progression from young to old very well, however, once we subject a system to interventions, it may not work. And this is exactly the example which is shown here. For example, Albertine, who is also a member of the consortium, I would say that I haven't mentioned that we also have many volunteers who help, for example, with the symposium and various other activities of the consortium, and Albert is one of them. So he has a manuscript, which hopefully will be accepted soon in Nature Aging, on causal biomarkers. And he identified changes which represent damaging changes versus adaptive changes. And each of them can be used to construct a clock, like, for example, damage clock based on damage, or adapt age clock based on adaptive changes. But when we apply this to rejuvenation situation, like here, converting fibroblasts to iPS cells, damage-based clock works really well. However, adapt age clock does not. And again, so this is the challenge, ongoing challenge that we work on. And also there's a conceptual paper we written with Mika Agrodnik on several examples of these adaptive changes. I also encourage you to read. Another challenge that we work on is so-called ratio between stochastic and regulatory changes. Because, for example, at the level of DNA methylation, as shown here, in the youthful state, the system might be, for example, fully methylated or unmethylated at the cytosine level. And then there could be co-regulatory changes, co-regulated changes when, for example, if one cytosine changes methylation, the other one would change similarly. Or could be stochastic changes when the changes would be completely random. Or, for example, mixed changes. So when both of these would be observed. So we try to analyze this at a level of bulk DNA methylation as well as single cell DNA methylation, and we clearly observe regulatory changes consistent with this model. However, we also observe stochastic changes when methylation happens randomly. So apparently, the situation is like this, probably in the middle. But what is the contribution of kind of programmatic processes versus stochastic changes is still unclear. And when the models built based on various machine learning approaches, some approaches penalize for the use of one or the other type of changes. And therefore, it could be reflected in the types of clocks that are developed. Again, this is an ongoing challenge and just one of them that need to be worked out. So what are the next steps? The next steps is a symposium, which will happen in less than a month at the Bach Institute, December the 4th, as I said here. So these are the speakers. We're very excited with the top leading experts in the field, both industry and academic researchers. So I encourage you to attend that symposium. It will be a part of Longevity Week. There are other events that happen before and after the symposium. For example, like Foresight, if you'd like to visit Foresight Longevity Weekend, it just precedes that meeting. So you can attend both. What else? We also prepare a first competition, actually, this Biomarkers of Aging competition, which will happen early next year. So what we've done, we profiled 500 human blood samples using Illumina methylation epic arrays, quantifying about 1 million CpG sites per sample. And this is associated with curated aging data from electronic medical records. And these are actually subjects from our MGB, Mass General Brigham Biobank at Harvard Medical School. And so we would encourage anybody to compete with your clocks and your biomarkers and see which biomarkers perform best in an unbiased way. Because currently, when papers are published, everybody claims that their biomarker is the best. Of course, every biomarker cannot be the best. But biomarkers can be used to quantify different aspects of aging. For example, we are thinking that phase 1 would be to predict age, and phase 2 would be predict time to death, because many of these subjects have died within 10 years or so since they donated the blood. And also predict aging outcomes, like, for example, multi-variability. And of course, this is just one step in the process. It will be a first competition, but hopefully would be followed up by other competitions. We are very fortunate that several foundations and companies, they help to support this. And I think Madi will mention about our sponsors in his presentation. So at this point, I'd like to stop, and maybe Madi would continue presenting on this consortium.
Adi, good to see you.
Thank you, Allison. Can you hear me well?
Loud and clear.
Okay. Is the slide in full screen mode?
Good.
Perfect. Thank you very much, Allison, for inviting me again. And great talk, Vadim. I'm going to follow on what Vadim discussed about the consortium, maybe discussing a bit more about our follow-up work after the cell paper and giving a bit more flavor about our competition and a bit of our plans coming up. Just a quick recap, my work with the labs, with the Snyder Lab and the Ladyshev Lab and the consortium mainly focuses on interpreting the biomarkers of aging, how to make sense of them and make them more meaningful, more interpretable, and to also move them toward clinical translation to address some of the challenges, some of the many challenges that Vadim just mentioned. And if you just Google epigenetic biomarkers or biomarkers of aging, there are already many products out there that you can just buy for direct to consumer use, and you get some estimates, some measurements of biological age, but it's very hard to interpret them currently. Their results are not actionable for the health, at least as far as we know in the science, based on scientific literature, not yet reliably validated. And the goal would be to validate these results to move them forward toward clinical translation. Just to give you a quick introduction, quick review of the basis of many of these clocks is, for example, here, one of the most common clocks or biomarkers, omic biomarkers of aging is DNA methylation clocks. And it's based on the idea of the genes, some of the genes that gain methylation or lose methylation very, very nicely with the progression of age. For example, here is the promoter of this specific gene. It's just one of the many genes that gain methylation by age. And you can see how cleanly, how nicely it's gaining methylation from 20 years old all the way to 100 years old. And there are many of them. If you put a few of them, a couple of them together, you can actually predict the age of the donor, the age of the sample with a very high accuracy. And there was also an observation that individual who have higher or lower estimate compared to the actual age, and that estimate, that difference can also predict their aging outcome. A lot of these omic-based biomarkers are based on this idea, but it's very hard to make sense of the results. Okay, as a physician, what can I do with this result? As a clinician, as a clinical trial person, how can I make an action based on this result? So these are some of the questions that we are trying to address in our own research and in the consortium research. This is an example of collaborative work that we've been working on for the past 2 years with the Snyder Lab group and with the Steve Horvath group and with the leading from the Vittorio Sebastiano labs working on epigenetic reprogramming. And the idea is to move away from these predictive markers to more biologically driven markers based on biological features of the genome and tie these changes in epigenome or other molecular biomarkers to underlying pathways so we can interpret them and we can start producing biological hypotheses based on these biomarkers. This is in addition to the work that Vadim just mentioned about causal biomarkers. In line with causal biomarkers, another approach would be to work on feature-based biomarkers. But besides addressing these technical sides of it, with the help of many labs in the consortium now, we're also trying to put together a roadmap for validation of existing and new biomarkers. There are other fields that already have very established frameworks for how to qualify a biomarker of disease, for example. But for the field of aging, we still don't have anything like that. From the experience, from the learning of our first work with the consortium, we learned that maybe the most important next step would be how to validate an existing biomarker that has been tested in a population to show that they perform well across populations, across diverse populations. First of all, we identified the steps that we need to take to reliably validate and replicate the result of a biomarker in multiple populations, focusing mostly primarily on omic biomarkers using blood samples, which is the most common approach in clinical trials and most non-invasive method. And besides that, we also put together a model for validation of existing biomarkers in a cross-population studies. And the idea is to move from testing one biomarker in one cohort to comparing multiple biomarkers in a single cohort, and then comparing multiple biomarkers across diverse cohorts to be able to benchmark them across a very diverse population and making sure that they are generalizable, the results. And just to study the state of the field, we also did a systematic review of the field, and with a very strict criteria, we identified the existing validated omic biomarkers in blood. And we had a few interesting observations. For example, as we expected, most of the validated biomarkers are already epigenetic ones. And then, surprisingly, multiple metabolomics and proteomic biomarkers have been also validated. And so far, we could not find a single transcriptomic biomarkers that has been validated with our strict criteria of around 10 years follow-up in an independent cohort, controlling for multiple factors and reporting hazard ratio for survival. So with these criteria, a bit of strict criteria, these are some of the prominent examples of validated biomarkers. And it's also surprising that some of them are now becoming very appealing. So some of them are validated in a large cohort of more than 10,000 individuals. Some of them have been developed using a diverse populations, around 26 cohorts here. And some of them are also reporting a very high predictive power. And these are all very promising measures that toward clinical utilization. For example, just to give you some context about this 10.9 adjusted hazard ratio, this is the paper that reports that number above 10 hazard ratio. And after adjusting for age, you can see that the stratifications of the top performing and lowest performing group is so much that in the top group, after more than 12 years, only a few percentage, only maybe 3% of the group pass away. But in the lowest performing group, again, after adjusting for age and other factors, around half of them pass away. And this is the stratification power that's very clinically relevant. And then this also happens to be true for diverse aging outcome. It's not just predicting cancer mortality, but also other mortality based on cardiovascular disease. So it's not just biomarker of a specific disease, but it could be considered as a biomarker of aging outcomes. Then there is also some studies by independent groups that already took the first step to benchmark the existing biomarkers. And again, for now, most of them are epigenetics, and they look at the multiple biomarkers out there and test their performance in a single cohort. And with this approach, you can start to have a comparison of their performance, at least in that population. So the next step would be now have these diverse biomarkers to be compared across population so that they are not so that we show that they are general for these populations. And these are all in our second work, in our follow-up review perspective work, and hopefully it comes out soon. We also identified, with the help of our diverse co-authors, our diverse members, we also identified some of the less utilized cohort and populations that has omic measurements, multiple omic measurements, as well as follow-up and aging outcomes that could be used in future validation studies. The other thing besides this kind of population validation is addressing some of the challenges in the validation process. For example, right now for the validation studies, every one of these biomarkers need to be implemented from scratch or using some of the public codes that's out there. And some of those steps are open to interpretation, or some of the normalization steps, some of the pre-processing that changes the outcome are not always faithfully replicated. So what we did is we collected all these public biomarkers, the algorithm of all these public biomarkers, and leveraging existing implementations of them based on the work of Morgan Levin's group and others group. We harmonized all of them into a single library, and this is an open library. Then you can already access it on this URL down here. And we are going to officially launch this at our symposium on December 4th. But this allows you to run any publicly available epigenetic clock at the moment with one line of the code and apply it to any properly formatted epigenetic data. And you can see here with 3 lines, we are running these 3 popular epigenetic clocks. And now we are also adding some clinical biological age to this bio package. And the idea is we set it up in a way that the community can come and contribute and add new biomarkers to this library. And this helps to have, moving forward, to have a standard format of biomarkers. And the other thing is to start benchmarking these different biomarkers. Now we already, in our library, we have more than 20 biomarkers, and there are many more. And we still don't know what the state of the art for their predictive powers for different outcomes. For example, for the age itself, predicting just chronological age, we don't know what's the top performing model, what's the minimum error that you would get for predicting age based on just blood samples, based on epigenetic data from blood samples, and how does that compare? How that compares with other omic? Are we already accurate at the same? Do we already have the same accuracy for proteomic biomarkers or other omic? So what we decided is it's easy to do this for epigenetics. We already have biospecimens collected many years ago, and have around 70,000 of them from our biobank. So we picked around 500 for a pilot study. Very diverse group of very diverse backgrounds, representative background, and as diverse as possible for age and gender. And already more than 10% of these individuals have passed away. So we know their aging outcome, and we have complete medical history, medical records for these people. And these are fully consented individuals that we can follow up. And we also have this live database to their information that is updated on a regular basis. The idea is to have this data as an ongoing benchmark data set to start comparing existing biomarkers, starting with their performance on predicting chronological age, and more importantly, predicting their aging outcome, including survival, time to death, and morbidity, multimorbidity. So we are still finalizing the design of this competition, but our goal is to start the competition, registration for the competition at the time of our symposium in December, and start in January. We have multiple phases, a few months for each phases, so that we can announce the best models, the best predictive models during the next year symposium. That's the goal. If you have any feedback, any interesting collaboration for this competition, let us know. And thanks for our fiscal sponsor, Methuselah, with tons of experience running prize competition, to also lead this competition, lead the operation of this competition. And if you are also interested to learn more about our work, past few months, we have given a few online talks that's been recorded, including our Foresight talks and our talks with our other groups from Healthy Longevity. These are all online, available for you to watch. I think 3 hours of talks that you can watch online if you're interested to learn more. With that, I want to thank all the labs that are supporting my work and the work of the consortium and our sponsors, Methuselah Foundation. And if Dane is in the audience, and I can share a few slides from his work. Has been our main fiscal sponsor, and also taking care of all the operations of the symposium. So it would be great if you can also hear from Dane about all the help that they are giving us and starting our partnership model.
Can everybody hear me?
Wonderful. Yes.
Yeah. Okay. So it looks like my deck is not going to be shareable unless I leave this call and come back. So I'll just go fast and we can just not do the slides. I did want to just dig in real quick, just a little bit more into the challenge structure in case there are folks here, because this is an amazing group who actually might want to contribute to the data science library that we're building, or would actually want to contribute to the challenge. Just to give a tiny bit more detail, we've, as they mentioned, we're going to have 3 consecutive tracks. They're going to run through the first 3 quarters really of next year, likely, on predicting chronological age, predicting mortality, and predicting multimorbidity. And again, it's really amazing that we're working with the Gladyshev Lab at Harvard, who has this incredible biobank. It really makes it a possible thing. There's no way that this would work without that sort of access. So we're incredibly grateful to be working with them. So we have these samples, which are going to make it possible for us to essentially check the models that people are going to be producing. We have 100K so far. And the idea is, as of right now, we're going to be splitting it 50, 30, 20 for first, second, and third place in around 30K. And then the third phase, which is around morbidity, that's the tough one, is going to be around 50K. And we hopefully will be able to award additional prizes to folks who, may have a really promising, interesting approach that the numbers might not quite be there yet for. It's good to be able to award innovation, even if it's not quite there, but to help encourage folks. As Madi mentioned, also, we've got this, the BioLearn library is really exciting to me, both because it makes it easier for us to do the challenge in terms of leaderboards, but it also allows for a diverse sorts of clocks to ad hoc be able to be added to a structure that can expand and adapt to really whatever kind of useful work people are doing in this field. So part of what we wanted to do with the challenge was both to induce people to join the field. There's a lot of amazing data scientists out there who are not biologists, but they know everything about AI and ML, so they should be doing this sort of work. We want to include them, but we also wanted to shine a light on the clocks that exist, put them up against each other, but also shine a light on the clocks that people might not know about. There's a lot of clocks that are behind closed doors, and we want to give them a good reason for them to be shared. The hope is really this is something that, opens the doors to a lot of folks and raises all boats and really presents the best of what the field has to offer in this area, so that there's more collaboration and co-opetition in that area. So there's a lot of work that we're doing to prepare for next year with several different companies who are in that sort of situation where they have really interesting clocks they're gathering their data from a variety of different methodologies. Vadim had a good slide in there where it talked about, obviously, you've got epigenetics and multiomics, and you've also got wearables. You've got a variety of different ways, physiological, functional biomarkers, all sorts of different things. And as time goes on, we're going to be trying to be more inclusive of the diverse ways of capturing all this data. And to that end, we're going to be working with those companies to ensure that the licensing structure that we have for both the competition and the library itself is very favorable and isn't going to make problems for people in a commercial sense. And so for 2024, at a quick glance, what we want to do is dramatically increase the number of clocks that we have in our library and expand the community. Again, pull in more data scientists to help us with this work. We want to take the epigenetic data set that we currently have and maybe bump it up by an order of magnitude or more. And again, we want to add multiomics. There's a number of companies that we're talking with about adding proteomics, metabolomics, other sorts of to the library. and then a whole bunch of cool stuff that we can't quite say yet. I also just want to say, and unfortunately I can't share this screen right now, but I want to thank the unbelievably amazing outpouring of support from all of the folks who are doing this, both the labs, right? There's no way this would have gotten started if it wasn't for Madi and Jesse and company and the Gladyshev lab and the support from Vadim and the Gladyshev lab and Snyder lab and Sebastiano and Eric Verdin, Andrea Meyer, everybody who's doing all this work. And we've got people in those labs who are volunteering to help out. It's really amazing to see. So thank you so much for that. And then also we have a number of different sponsors, which is really cool. Some of them are familiar and others are actually completely new to this field. So that makes me feel really great. So the Volo Foundation, they are very data science focused. They're very cool, lovely group of people. Navigage Foundation as well. The Buck has been very supportive of us and they're kind enough to host us in 3 short weeks. Hevolution was kind enough to support us there through the AFAR conference program that they have. Quadrascope, I see Fiona's in here, so shout out to her. Thank you so much. She helped us out like big time when it came to doing the epigenetic testing on these samples. So she saved our butts there for a little bit. Hey, she popped up. Hey, thank you. And Olink, really cool proteomics company. Foxo did the epigenetic testing for our initial challenge, so we're very happy to have their partnership. Gero is great. They're new to the field. They have a very interesting approach. They're very cool. They have good clocks. BioAge, the ubiquitous VitaDAO. Thank you, VitaDAO. And Open Cures, a good old friend of mine, Kevin Parrott, who was with us at the beginning of Methuselah and has a really cool platform. And then there's probably going to be some others. I also want to say thanks to everybody who's actually going to be coming to the symposium. Happily shocked that we actually sold out today. So we hit 200 people, which is pretty wild for a nerdy conference like this, with no pitching allowed. And so we've got 200 people. We have, I think, if I recall correctly, 23 posters already. And so we're super excited about this. It's really crazy. And so for anybody who still wants to come, please do sign up. We have a waiting list. If other people end up canceling, we'll do our best to get you in. So that's my spiel. If you want to contribute in kind financially, you want to send something, or you want to add some data or a clock or some sort of tool to our BioLearn library, if you want to compete in the challenge, please reach out to myself or Madi. And we can throw our emails in the chat, and we'd love to talk to you. So that's it. I'll stop talking. Thank you very much to everybody.
Wonderful. Thank you on the tons of updates that you guys have. Really exciting. Congrats on all the hard work that you've been doing, and thanks for your hard work. It's a lot of herding cats, and we appreciate all the efforts that came from you guys. Okay, we have tons of questions already. Do you mind if we just start with a few, and whoever of you feel inclined to answer them, please just jump in. First one, I have Paritosh. If you want to unmute, the stage is yours to ask your question.
Hi, thank you so much for this awesome presentation. And I deeply enjoyed knowing the drawbacks and bottlenecks. I think that's the most important piece as we go into the next stage of development in some ways or the other. I had a question around a table which was just before the BioLearn slide. Couldn't see the slide number, but could you please explain the significance of the figures in that table, Mahadi?
Let me see which one was that. So this will be recorded, and it's already recorded, and I think it will be hosted on Foresight channel. But besides BioLearn, yeah, we have a list of cohorts that are identified. So we actually made it open to all the members of the consortium to vote on their favorite cohort to be promoted in the second paper. It should be not utilized. It should be less commonly used for biomarkers validation. So we all know Framing study or a lot of top meta studies. So these are the popular cohorts. But we also know that there are some newer cohorts or some less commonly known cohorts that are ideal to validate or build new biomarkers. So we invited everyone to vote on their popular cohorts, and we collected all these votes with the information, with the notes and their justification. And we came up with the top 10 cohorts or population, biosphere population that could be used to test the existing biomarkers and benchmark the biomarkers. And UKB is one of the most popular ones that are still not utilized as much for testing omic biomarkers of aging, because they are still producing those omic data. So UKB was one of them. Our own Mass General Hospital Biobank was voted as one of the promising cohorts that are not as much utilized. And then some other ones. So we picked the top 10 that the consortium thinks could be used to validate future or existing biomarkers moving forward.
Wonderful.
Thanks.
Thank you so much. Next one up, we have Priyan.
Thanks, Allison. Okay, so there's a lot of clocks, and they're not just ohmic clocks, right? There's some simple clocks like blood glucose variability and grip strength and really simple ones like that. And by the way, with those, we know how to reverse them to some extent or improve them, right? So my question is, with all this emphasis on big data and AI and omics, let's say that you found the great clock, like the really killer clock that shows you biological age and particularly shows you when it's departing from chronological age. So if you know if someone, according to these measure, is aging fast or slow, now what? You can get all this data, you can run all these statistics, you can find the world's best clock or the right ensemble of clocks. Why are we doing this?
I know Vadim had a good answer to that. Do you want to go ahead, Vadim?
I suggest that I give a floor to Jesse, who is also a member of the consortium here, but he hasn't had a chance to present. Jesse, would you like to answer?
Yeah, I would say because it's one of the clear steps to bringing longevity interventions to people to improve aging and ultimately their lives. We know we're not going to do lifespan studies in humans. And so these tools, and it seems to be the case that these big data omics-based tools are very powerful. These tools offer a roadmap to be able to do that and to validate longevity interventions. That's what I would say.
Okay, so let me see if I understand that correctly. So what you're saying is because you can't wait for people to die, or that's undesirable and takes forever, or it takes a long time, you use this as a proxy for whether a given intervention is actually improving their predicted lifespan?
Yeah, that's the basic idea, I would say.
Okay, thank you very much.
Yeah, it might be worth, I'm not the right person to talk about it, but whoever wants to talk about surrogate endpoints, it might be a good thing to spend a minute or two on. Anybody? No.
The basic idea is that we just said lifespan is not, or longevity is not a good endpoint, a primary endpoint. And so the basic idea of a surrogate endpoint is it's something that stands in for a primary endpoint. And so here again, using a predictive tool like one of these omic biomarkers of aging would be a surrogate endpoint for a clinical trial to test the longevity intervention. Hope that was brief enough.
Okay, but at some point you have to actually verify that you're not just having a correlative thing and you're treating the symptom by unmethylating or whatever it is you're trying to do, because you don't know that changing the clock is going to change the lifespan until.
Absolutely. So that's why we place a huge emphasis on validation in our current work. And as a big picture research question, as Vadim highlighted, the differences between causality and correlation and aging are very important areas of ongoing work.
Last thing, I'll just say one of our good friends here said that correlation isn't causation, but it's correlated with causation.
All right, wonderful. So Meta, you're next, and then we're hoping that we get just your hand raises as well.
Cool. Thanks. First of all, great presentation. I'm excited about all the work that you guys are doing. Hats off. One of the questions that we had, especially not only me, but like at True Diagnostic, we found through some of the analysis that the Epic V2, they did remove a lot of those probe sets based on multi-mapping. However, a lot of those probe sets were also very important in predicting a lot of the Dunedin pace and additional clocks as well. I was wondering, there isn't a variant of the Epic V2 which includes the age add-on chip, so the additional CPGs that were missing. Is that something that you guys have been thinking about, running those 500 samples that are being used for the clock challenge? Just because it appears that even though they are multi-mapping probes, they're still relatively important for some of these very predictive clocks.
Yeah, that's a great point. We've been discussing all these challenges because a lot of these epigenetic biomarkers have been developed years ago with older versions of microarray assays. But at the end, we decided to go with the latest technology that's been advised and promoted by the company Illumina, who is building this. And a lot of those slides have been removed because they had some issues So we decided at the end, go to the version 2 EPIC. We already sequenced those with the help of one of the leading companies who is producing these all in one batch. So it's probably the cleanest data that I've already seen out of hundreds of data sets. So on the quality side, this is probably the highest quality that we can get. That's also fair, and that's also future minded, because now it's the state of art. The older technology is being depreciated. So that's the state that we have. But at the same time, the idea is this is a pilot study, first year, just as a proof of concept. We want to show that this is doable, this is feasible, and then promote this, like many other fields, promote this yearly benchmarking through competition with more sponsors, with more omics. That's the idea.
Maybe I would add that although this initial set is based on this Illumina array, we're also interested in sequencing these samples, and therefore like additional clocks could be used in additional methods. So we're searching for additional sponsors so that we could extend this data set not only with regard to technologies, profiling technologies, but also the number of samples for future competitions.
Also,
we currently spend some of our own money just to help the community with this data set. So hopefully other people could contribute as well.
Oh, okay. Let's see how many others we have time for. Abdul Kader, you're next.
Yeah, thank you. I noticed from one of the slides that was mentioning of the value of peripheral blood in detecting aging. So I was wondering, how can you explain the validity of this tissue as an accurate measurement and predictor of aging? Can you explain that to us?
Maybe I can start, and other people we express our opinion, right? So nobody knows ultimate truth here. But to me, blood is a very good tissue to use, simply because it's quite stable. It interacts with organs, and therefore it can be drawn in a longitudinal way. Of course, it has limitations. It might not represent aging of some tissues in the body, but to me, it's currently the best available.
Yeah. Do you think there is a special consistency for an organ or tissue not to be represented in a blood proxy measurement?
This is still unclear, but there are different types of clocks that can be used based on the analysis of the blood. For example, Levin's lab or group develops a systems age clock where they train the clock to quantify aging of particular organs based on the blood samples. So there are other ways that other people are developing. We are also working on that in our lab, but which is ongoing work. So it's ongoing studies. Hopefully it will become more clear in the future.
Yeah. I think it's a great work that you do that. Thank you very much for your efforts.
Perhaps final question from Keith.
Sure. Yeah. I just wanted to speak to some of the challenges that Creon was bringing up earlier. This is one of the reasons why having multiple different effective clocks is so important, because then you can tease out causality. As an example, maybe a therapy has a health span effect, but it might actually be because it's making the person more peppy and wanting to exercise more, which is different than something systemic. But if we had a physiological based clock and a deeper clock, that might be able to tease out what's actually going on in those cases. And I just also wanted to underline one other bright spot of another value of even simple physiological clocks is motivation, right? There are projects out there, Michael Gear and Peter Ward in Humanity Health, that are showing that even if you present someone with simple data that shows there's a deviation between their chronological age and biological age, that's enough to motivate them to actually take action. So I think that's another answer to the question of why is this valuable?
Wonderful. Then with 1 minute left, are there any final bits and pieces that you want others to know about? We already discussed, and I shared here in the chat, the Biomarkers of Aging Symposium. If you're watching this afterwards online, you can definitely check that out by just simply Googling it and perhaps join the wait list. So we'll see if we see you hopefully in December. Anything else that I've been missing to any of you presenters?
I forgot.
We'd like to thank everybody for your support and interest in this, because this is, again, so I want to stress it's a community effort. It's not just our kind of groups, right? We want to help and work together to develop this for all of us, for the humanity.
Yeah, and so far it's really felt like a big community push. So it's been really interesting once you put something out there, how many people collaborate and chime in. So thanks everyone who has collaborated, reached out, cold emailed over the past few months. And I think it's really off to a really great start, and there's so many exciting projects that you all can participate in. So I hope that you continue to do so. Thank you so much, everyone, for joining. Thank you guys for presenting. Really exciting stuff. Feels like the work is just starting, really. And yeah, I will hope to see you all in person in December, either at the Vision Weekend and or hopefully at the Aging Consortium Summit. So see you all very soon. Bye-bye, everyone.